Yeah, I'll introduce Dina. Dina is my PhD supervisor, and Dina is also a staff scientist at the Max Planck Institute, and she sits in Möggingen. Dina is a behavioral ecologist, but she also is really interested in evolution. And she's mainly working with small mammals, better said, shrews, but all different kinds of bats too. She focuses mainly on fluctuating resources. That means resources which are not available all the time, but they fluctuate in time and in space. And she then investigates how animals react to that. So not only in their behavioral, like social foraging, or how they find the flies, for example, but also in their physiology, how they adapt their body to then if maybe animals would starve because they didn't find the resources. She doesn't only do research in the temperate zone, but also in the tropics. So she has collaborations worldwide. Sorry, can you repeat? I heard someone talking. I will just go on like that. So yeah, she has collaborations worldwide. She has worked in Panama, in Portugal, in Poland, Spain, Denmark, the US, and also in Africa. And today she's presenting her research which focus on bats and how they adapt to the fluctuating resources. And we'll give you an overview about the findings of the last years of research. I'm really looking forward to it. It's always exciting to see what Dina is doing, not only my project, but yeah, all the stuff she's doing. So yeah, really looking forward. And so I welcome Dina to present her research.
Thanks very much, Lara, for the lovely introduction. I will share my screen now. Can everybody see my screen? I'll just assume so. So welcome to my talk. This is my first ever virtual talk, so I'm a little nervous about this, but I'm sure it'll go well. And yeah, I'll give you a bit of an overview over my research, focusing on the ephemeral resource adaptations. Before I start into the talk proper, I always like to start with acknowledging that this is the product of many people's work, not just my own, and also different funding agencies. And I want to thank especially my lovely group of all women currently, who are wonderful to work with, and some of whom I will highlight during the talk because I'll be presenting also their work. Okay, like you saw from the title and from Lara's introduction, I'm interested in resource ephemerality, that is how resources, mainly food, a little bit also roosts, are distributed in time and space, and whether they're predictable or not, or whether they change in their availability over time or not. And then how this can create bottlenecks where resources are scarce, and the adaptations that animals have developed in their behavior, physiology, and morphology to deal with this variable and unpredictable resource landscape. So to give you an image of that, if you imagine that the animal I study is this barrel, then this animal has to maintain body heat, which is always a bit of a source of loss of energy, which is exemplified by this dripping tap. It has to behave, move around, and it has to maintain its body, grow tissue, and reproduce. And all of this has to be fed by energy that it gets through foraging. And so if foraging becomes difficult and unpredictable because of the resource landscape, then all of these processes become endangered. And I'm interested in how animals adapt to that. One red thread that goes through my talk is that even though I build a lot on findings from the lab, and I also do short-term captive experiments sometimes with my animals, I really believe in looking at the animals in their natural environment. I'm interested in evolution, as Laura said, and how selection acts on my animals to produce the current phenotypes that we find. And so I do almost all my research on wild free-ranging animals, which sometimes affects the amount of work I have to do as well as the sample size a little bit, but I really believe it's worth it. And the other thing that is a red thread through my work is that I work on small mammals with very high metabolisms, especially shrews and bats. So if you look at this classic representation of body size against energy consumption, then the animals that I work with are up here. They're like through the roof, tiny little animals with really high metabolisms. And because shrews are predominantly solitary, today I'll mainly talk about bats, as I assume that you'll mainly be interested in how this all is connected to my work on social behavior. So like I said, we're interested in the distribution of resources in time and space. So I want to give a little bit of an introduction and some examples for that. And we are interested in this at several levels. So resources can be variable or unpredictable in space, but also in time. And they can do this at a broad scale, such as over the year, think of seasonal changes in resources, or at a more fine scale, such as the occurrence of flowers on a certain plant during just a few days. And one example that we work with a lot are insect swarms. So as you can see in this video, this is really typical how insect swarms can be super abundant, but spatially extremely localized. So this is a huge amount of insects, but just a few meters to the left and to the right of it, I hope you can see it, there's just empty space. And that as we work on on bats that are specialized in swarming insects so much, we invest a lot of effort in quantifying this resource and showing the ephemerality of it. And one big project where we have been done that is already published is monitoring of insect swarms in Poland over the course of several summers with camera transects, where the cameras point at the sky and take flash pictures every 5 minutes, and then you can count the insects on these pictures. And the main finding that we found was that 99.999% of the pictures were empty, not a single insect on them. And then a few more had just 1 or 2 insects on them, which still is not a resource that's abundant enough for most bats to explore. Flying to find these insects would cost more than they would get out of eating the insects. So we were really interested in the occurrence of insect swarms. And we found insect swarms on really just a small percentage of the pictures, and they showed very clear patterns. So over the course of the year, here you see the whole course of summer, clearly insect swarms peak just around June and July. Over the course of a single night, we also see clear patterns. So insect swarms occur just after sunset and just before sunrise for a short time, and even in between, there's really nothing available. And then, oops, I can't really see this. If you look across different habitats, there's also a clear bias towards habitats over water that have a lot more insect swarms. So it is a very ephemeral resource, lots of empty space, and then even where the insect swarms are is highly variable in space. Oops. So if you go back to my figure from before, for insect swarms, how do they fit in here? They are spatially fairly predictable. They occur over water. Temporally too, they occur during June and July. This is the rough scale, but if you look at the fine scale, still temporally pretty predictable, just after sunset and just before sunrise. But spatially, where over a given water body these insects will occur is highly unpredictable. So potentially, if you can somehow increase efficiency of finding this resource through social behavior, for example, then that will be a big advantage. Another resource that we work on a lot in Panama now, Laura mentioned that we work in the tropics a lot too, are fruiting fig trees. So this is a map of all the fig trees around our study site in Gamboa, Panama. And they go from small and green, which means fruit just starting, or from gray, which means no fruit, to fruit just starting. And then as the redness and the size increases, they ripen, and only the red figs, the red circles are ripe fruiting fig trees. So fig trees are a bummer because they don't fruit regularly, and they don't fruit seasonally. So a given fig tree may not fruit at all in one year, and then maybe fruit twice in the following year. So for each given fig tree, it's unpredictable when it will fruit. And this creates a really uneven resource landscape. If I play this again, there are times when there's lots of big red circles, and then there's times like now when there's hardly any resources available. So not only is it spatially unpredictable, but there are also bottlenecks. And if you look at this now again, in context of my concept here, then the figs are now unpredictable at the broad scale. We don't know where the next fruiting fig tree will be, and also not when it will fruit. But at the fine scale, once a tree fruits, it's not going to move around, it's going to stay in place, and you can return to it. And also the resource lasts for about a week and is very abundant. So here at this scale, a bat might have a problem finding this resource. So now let me give you some examples of how this all ties into adaptations by bats to in terms of morphology and so on. So let me start with morphology. And here we have this really nice advantage that bats have wings, and their wing shape tells us a lot about them without us even having gone out a single night and following them. Because similar to birds, the wing shape is reflective of their adaptation to where and they move in three-dimensional space. So here, at the one extreme, you have a very narrow winged bat species, has long, slender wings and a low surface compared to their body weight, resulting in high wing load during flying. So these bats have energetically costly flight, even more costly than flight is anyway. And they're not good at maneuvering, and they're actually incapable of maneuvering in narrow space, but they fly in the open air fast and far. And then as you go towards increasingly shorter, broader, and rounder wings, edge and gap foragers or water surface foragers, you finally get to the other extreme, which are very round winged bats with large wing surfaces that are highly adapted to navigating in the understory, flying around leaves and trees. They're good at hovering and at flying slowly. And their flight is relatively less costly, but they can't fly large distances. So we use this concept a lot throughout our work, especially for comparative studies. And I want to give you just one example here of how we can learn something about brain size and how it's adapted to the ecological niche based on that. The brain is one organ that I'm particularly interested in, especially also in my shrew research, because not only is it a very important organ, but it's also energetically very costly. And luckily, it also varies a lot among bats, not only in terms of morphology. So these are different brains of different bat species, but also just in absolute size. And if you now plot this wing shape area, which is one really important wing parameter that I just talked about, corrected for body mass against relative brain size, and you look at the different gilts that I just showed you, then you can see from the open aerial forager to the understory, to the edge and gap forager, to the water surface forager, to the understory bat, from narrow wing to broad wing, you have an inverse correlation with brain size. So the open aerial foragers have comparatively smaller brains than the understory bats. Expensive flight, less expensive flight. And if you now, interestingly, if you split this up between bats that eat fruit or nectar and bats that eat animals, mainly insects, but also sometimes other animals, you can see that all this variation in both wing shape and brain size comes from animal eating bats. And that makes sense because if you think about where their food is located, then all plant eating bats pick up their food from plants by definition. So they have to be able to maneuver in the tree and be good at that, whereas animal-eating bats can find their food anywhere in the open air, such as swarming insects, to edges and gaps, surfaces, or within the tree crown, like a katydid sitting on a leaf. So there's a tight link between the ecological niche and thus the food bats eat and their brain and wing morphology. And possibly this is also tied to energetics. So along these lines, we were then also able to show or reconstruct how this happened in evolution, this changes in brain size. And that was very cool to find because it had not been shown before. That is, we reconstructed the ancestral morphology of a bat. And in terms of brain size, body mass, and wing area, this bat was intermediate, probably lived in the forests and foraged along the edges and gaps. And then from this ancestral bat, with a medium-sized brain, in the course of evolution, brains got bigger, but also smaller when selection acted on them. And that was almost impossible to publish at the time, because at the time the belief was whatever you gain in terms of cognitive abilities, you don't give up anymore, because a bigger brain is a better brain. And really nobody wanted to believe that brains could also be selected on to become smaller again. So still a finding that I'm fairly excited about. Okay, so that was just one example to show how like this ecological niche and diet and morphology can be connected. But now I want to give you an example of how this all ties in with physiology then. And for this, I want to use a bat as an example, which is specialized on the figs that you just saw the map of. This is a project that I did with T. Gomera for most of the time. And these cute little guys, Uroderma bilobatum, are highly specialized on these figs. So they feed on a resource that is unpredictable in time and space. And at the same time, this resource, figs, are fruit, and they don't even just eat the fruit, but they just squeeze out the bits of figs and swallow the juice and spit out the pulp. So they basically feed on a diet of fruit juice, which should be energetically fairly limiting, because sugar water is not a very good way to source for storing energy. And also because these bats have an expensive mode of locomotion, flight. It's about 15 times more expensive than sitting still. And they have to fly out in the evening after not having eaten all day on an empty stomach before they can replenish. So we wanted to know how quickly then, when they start flying out and find food, can they start to metabolize it and reboost their energy. And to do that, we caught them and having fed on their natural diet of figs, which have a certain, they're C3 plants, so they have a certain carbon dioxide predominantly in them. And then we switched their diet to a C4 plant, agave syrup, and collected their breath as they exhaled it, and measured the ratio of carbon isotopes in their exhaled breath. And from this, we were able to tell that just freshly ingested food is metabolized within as little as 8 minutes. So that means as soon as these bats eat, they can start to use this resource to power their flight and their life. Imagine if we could do that, we would not have such a problem with marathon running anymore, because we could just keep replenishing. However, it's good that they can metabolize their food so quickly, because they need a new energy source in the evening, but this also translates in really fast energy turnover. So the T50 for turnover of their entire body fat is just 1 day. So in 1 day, they have turned over 50% of their body fat, which already puts you in trouble if you don't then replenish soon. So having found that, we were also, of course, interested in how much energy they then spend during their daily lives to balance this. And to do this, we use the technology that we use a lot in my group, and that's little heart rate transmitters, which emit a constant frequency signal, and they have 2 little electrodes that go under the skin and monitor muscle potentials and transmit this as sound. And from the sound file, you can then extract these spikes, which are the heartbeats. And when we attach the transmitter to the bats, we first put them into a respirometry setup and measure their oxygen consumption at different heart rates, and then release them into the wild and follow them. And found something really amazing. So in terms of flight energy expenditure, nothing much surprising. energy, the cost of flying is high, so energy expenditure is high. Also during the night's rest, nothing much surprising, about 400, between 450 and 500 beats per minute is what you would expect from a bat this size. They're about 14 to 16 grams. But then when we analyzed data from daytime resting, we found a big surprise. So on average, still not very surprising. It's a little lower than during the night when they're still probably a bit more active, but kind of in the range of what you would predict. But then there were these downward spikes. And when you consider how much difference these downward spikes make, it was 10% of the daily energy expenditure. So, oops, can you see my mouse? Normal energy expenditure during flight as expected. Energy expenditure during night rest as expected, but then much lower than expected heart rate during the night. So somehow at these high ambient temperatures that don't let them lower their body temperature like bats in the temperate zone would do in order to pull along their metabolism and save energy, these guys stay normothermic, maintain their body temperature, but are somehow able to still lower periodically 1 or 2 times an hour for a short time their heart rate and save a lot of energy that way. And this is a really important finding also because normally when heart rate transmitters were used before, including myself in my first heart rate study, because it's so much work to analyze these data, we only analyze 5 seconds every 2 minutes or something like that. And then everybody would discard these values as outliers. But because Teague and later Lara have developed a method to automatically analyze these data with R, we can now analyze nonstop data and uncover these really cool strategies. So Uroderma moves food into the metabolism quickly, which they need because they have this poor energy source. But this also leads to rapid fat turnover, which they partly compensate with this bradycardia, where they modulate the heart rate flexibly to save energy. But still, energy expenditure is high. And here's my first little sneak peek from my group. So Lara, who introduced me, is now working on this in a temperate zone. Remember, I just said bats in a temperate zone can use torpor. So they will choose a cool roost, which allows them to lower the body temperature as low as ambient temperature. And then that will also pull along the metabolism and allow them to save energy. But reproductive bats, females that are pregnant or lactating, and males during spermatogenesis avoid using torpor because it slows down these reproductive processes. So Lara is looking at whether European bats will also use similar strategies to save energy during those times when they want to stay normothermic. So what you see here is body temperature that she measured subcutaneously and with as the device on the skin. And this kind of goes along in a pattern with ambient temperature. And you can see heart rate, which stays mostly high in this male during spermatogenesis. But there are some really cool things going on that kind of look like the bradycardia I just showed you. But this is just a sneak peek, and I'm sure Lara will tell us more about these patterns soon herself. So after this one example of how the physiology is adapted to maybe or partly compensates maybe for poor resource availability or ephemerality. Let's get into the part that probably interested you guys most, and that's social behavior, social foraging. And the background here is that when I started working on bats, I was actually interested in why they are social. And I very quickly started thinking about why then tropical bats are social, because the going theory was based on work from a temperate zone. At the time, most work had been done in the temperate zone, where bats are only social in the summer. People assumed that the costs of being social outweigh the benefits of being social for bats, and that's why they're only social, the females are only social, or only the females are social in the summer when they're pregnant and lactating, and they can't use torpor, so they'll live in groups so they can thermoregulate with each other and save energy that way. And then I wondered, why would so many tropical species be social then? Because temperature shouldn't be such a problem, but all the costs like parasite transmission, competition, and so on should still apply to sociality. And that's when I started thinking about the structure and availability of resources, mainly based also on some work by Kamran Safi during his PhD on male sociality in temperate zone bats. And that was to add to all these parameters that are really important for social foraging, like Stephen and Damien showed in their nice review a few years ago, and which also play a role, of course, for bats. In addition, maybe social foraging, so socially looking for food in some way or other, could be important for bats that are specialized or dependent on an ephemeral resource. So if a bat flies out in the evening to look for food, it is really crucial whether its resource is evenly distributed in time and space. So if it eats a food, finds and eats a food item, there's no more food nearby. And that means it's not useful for a conspecific to follow it and or coordinate its flight with it, because there's nothing to share. But if the resource is ephemeral, such as the insect swarms or the figs that I showed to you in the beginning, then this food source might be difficult to find. Remember, 99.9% empty pictures. But once you find the food, it's clumped and you can share it. So this is a concept that we think and talk about a lot. And Jenna Coles, my PhD student, is actually just currently working on a review on that to look at how the properties of the resource, whether it's dispersed or not, whether it's spatially and temporally predictable or not, and how long it lasts, like just an hour or two, like the insect swarms, or over longer time periods, and then link to that what kind of social information bats use and how long lasting this information is, how all of these parameters work together to translate into the different foraging strategies that we see in bats, from fully solitary to group foraging and all the other steps in between, like local enhancement, following recruitment, and so on. So on the one side, we have the resource properties, and on the other side, we have the bat properties. Like, is predictability low or high? Is it available for short or longer time periods? And can it be shared? And then for the bat, how strongly specialized, and kind of linked to that, how dependent on it is it? And based on these parameters, we then make a prediction about when, where, and how information should be transferred. Can it be transferred with a long-living cue like smell, for example, or is it a short-lived cue like sound? And can it be shared in the roost, or does it have to be shared at the foraging site because the resource lasts so short? But let me give you some examples again. So the first example, again, is the fig-eating Uroderma bilobatum. Remember, ephemeral figs are their food source, unpredictable in time and space. However, when they are available, they stay in the same place and they're available for several days. They're huge, those fig crops on the tropical trees, so that's a highly shareable resource. And the energetically limited Uroderma with a high energy expenditure, in spite of some maybe modulating strategies, are very strongly specialized on figs. They don't eat anything else. And that means because also of their high energy turnover and so on, they really strongly depend on finding this resource regularly. So we thought that if this resource lasts so long and a bat eats these figs and then returns to the roost, it would bring a cue with it, the smell of the ripe figs, that other roost members could then use, especially those that did not know where the next fruiting fig tree was coming up, and follow it using passive olfactory cues. And when we looked at them a little bit in more detail, so yeah, they have this fast energy incorporation and high energy turnover. I've said this several times now, and they have compensating heart rate strategies, but still depend on regularly and reliably finding their food. We also find that they have a bit of a fission fusion society. We're also interested in how these foraging strategies affect social stability. So not super stable. And we thought that social foraging in these bats would probably be rare. Like only if I don't personally know where the next fruiting tree is coming up, I will need this information and follow my group member. So it's rare, but really maybe crucial. And we did some experiments where we tested this in short-term captives, and we were able to show that they use these olfactory cues of the smell of food on their conspecific breath, not only as information, but also they evaluate the quality of the information. They can distinguish between whether their group member has eaten this food source or just encountered it. So it seems like they really may use passive information exchange in the roost during those rare times when they don't know where the next food source is coming up. So in this bat specialized on the understory, we think that the social foraging strategy that they use is following. Another example now is a bat that uses the insect swarms that I introduced in the beginning. This is Noctilio albiventris and is a water surface forager. As you may remember, this is kind of an intermediate phenotype between narrow-winged and broad-winged. So this bat has high temporal predictability of its resource, remember, just after sunset and to a lesser extent just before sunrise. But the spatial predictability of this resource is very low. We don't know where over the water in a given night these insects will occur. And crucially, this is a very short-lived resource, just for an hour or two, and then it's gone until the next night. But it's abundant and shareable. And these bats are fairly flexible in the wing morphology, so we expected that when they're foraging for insect swarms, they probably would use some sort of social foraging strategy to find it more efficiently, but they could also use other food sources, and thus they would also not be super strongly dependent on it. But we predicted that when foraging for insect swarms, that they would use passive information that is contained in the echolocation calls that they inadvertently produce while looking for their food, and that other group members can use to gain information. So this would be information transfer on the wing during flight via sound. And the way this worked would be, so imagine a bat flying, sending out search phrase calls, looking for insects, and it can detect its typical prey size from about 4 to 5 meters in case of Noctilio. So that's not very far. So you have to fly around a lot to find the dispersed resources. But the same bat, as it's calling, can be heard by its conspecifics over 10 times that distance. So this bat can hear this bat calling and producing search phrase calls from 40 meters away. So if this bat now finds a food source and switches from search phrase calls to feeding buzzes, which are higher rate and differently structured calls, it can learn from this switch that the first bat has found a food source and go and exploit it too. And this is what we thought would be happening in these bats. And we went out and tracked them to check. So what we find what confirmed all of these hypotheses. So we also looked at breath isotopes and energy turnover. And similar to the fruit eating bats, they have very fast food incorporation and high fat turnover. The groups are harems, with the females living in fairly stable groups, the males changing a bit more often. And astonishingly, these bats are only active for about 1 hour a day. And during this 1 hour, and I had a very hardworking student count insect heads in the droppings that a bat brought back from foraging, that corresponded to 700 insects. So you have to be really highly efficient to catch that many insects in 1 hour. We then group tracked these whole groups of these bats and found that they did group forage, but only seasonally, when the females were pregnant or lactating, which actually overlaps for a while because they have 2 birth peaks in a year. And they did it about 85% of the time during that time. And 85% is just because we sometimes lost contact with the bats due to the range of our antennas. And then we also showed with playback experiments that they do use passive acoustic cues, like that switch from search phrase calls to feeding buzzes, to go and find conspecifics that they think have found food. So they have Yeah, so these intermediate bats with a kind of intermediate dependence on this resource, or let's say a seasonally high dependence on this resource, would use coordinated group foraging to fly to a feeding patch. And here's another sneak peek. So this is Jenna Coles, my PhD student who works on Noctilio in Panama now, because now technology has advanced sufficiently that we don't have to use radio transmitters anymore, but we can put GPS tags on them. And so she's GPS tracking them, and hopefully soon also adding acoustic transmitters to find whether they group forage. This is unfortunately data from this time period when we would not expect them to group forage, and they also don't, except sometimes you see some hints for it too. And she's also monitoring insect availability at the same time at the study site. So again, keep your eyes out for more information from Jenna directly. Okay, so now let me give you a last example of a highly specialized bat that is completely dependent on an ephemeral resource, and that's Molossus, the Pallas free-tailed bat, which is the other extreme of the morphology spectrum. So very narrow-winged, one of the most narrow-winged species. It's like a swallow or a swift of the night sky, and it cannot, if there's no insect swarms that it can find, it cannot switch to foraging along gaps or edges or in gaps or something like that because of this wing shape. So it eats insect swarms or it eats nothing. And again, temporal predictability of this resource is high. It's the insect swarms. Spatial predictability is low. These insect swarms occur somewhere else every night. Its availability is low, but its shareability is high. And this bat, because of its morphological adaptations, is very highly specialized on insect swarms and also very highly depends on it. So again, we expected acoustic information transfer using the same mechanism like I showed for Noctilio during flight in the foraging arena, coordinating the flight and staying within hearing distance, but we expected them to do it all the time. And when we went and checked, we found that they were active on average even less time per day, so only 37 minutes per day, which might be reflective of the fact that flight is so costly for them. They have to really spend as little time as possible flying, but that also means that they spend 23 hours a day or more in their roost, fasting and doing nothing. We found social foraging of the whole social group here. They also live in harems, but the males forage together with the females. They're also dependent on this resource all the time. And we found group foraging at least during 70% of the time, but again, these bats have a big home range, and the 30% are times when we just simply didn't hear the bats. They do probably also use passive acoustic information transfer. We have not shown this for these bats yet, but other people have showed use of passive acoustic information for bats in this family. And the groups are small and extremely stable. Basically, once a female forms a group with other females, she stays there until she dies, and she never leaves the group, no matter how much you disturb them, for example. We also studied their heart rate and found another strategy different from the one Uroderma uses. So these bats roost at high temperatures in the roofs of buildings, which get really hot, so they cannot lower their body temperature. But somehow they are able to uncouple their heart rate from their body temperature and lower the heart rate independently for extended time periods. So instead of 400 to 500 beats per minute, at least, this is a 10-gram bat, they can lower it to an average of 140 beats per minute. That's incredible for such a small animal. And they can go as low as 40 beats per minute, while all the time their body temperature stays high. And what we have been able to show in these bats, and this is again Jenna's work during her master's thesis, is that they also use information contained in the search phase calls. So they can recognize group members from their search phase calls, which probably helps them to coordinate flight and staying within hearing distance and not accidentally following a different bat that might have eaten more already and does not have the same needs to meet as your own group members. So in this highly narrow-winged, small-brained, open aerial forager with strong energetic limitations, we also found group foraging, but all the time. What's really cool is that there's more and more evidence accumulating that supports these ideas, especially a lot of that work coming out from Yossi Yovel's group in Israel. Yossi was actually a person who was very skeptical about this in the beginning. I mean, that first paper about passive acoustic information use in Noctilio was probably my second hardest paper ever to publish, because nobody wanted to believe it, that something as strongly selected on as echolocation calls would contain social information. But it's a very commonly accepted idea now, and Yossi and his group in one recent paper compared 5 species that have different resource ephemerality, and were able to show that social foraging, the degree of social foraging increases with increasing ephemerality of the resource the bats use. And the nicest example here is the Mexican fish-eating bat, which Edward Yermy studied during his PhD. Edward is coming here to join us as a postdoc soon, hopefully, once the embassies open up and he can get a visa. And he tracked these Mexican fish-eating bats with transmitters that record GPS and sound, and looked at whether, as the bats fly and forage for swarming fish that come up from with the cold waters in this deep water trench, there would be calls of conspecifics recorded on this bat's transmitters, and whether these calls would then, or the frequency of these calls would be increasing when the bat carrying the transmitter was producing feeding buzzes and thus feeding on something. So, and as you see, indeed, each of these bats, as it flies during its nightly trajectory, is always within hearing distance of conspecifics, and especially during the yellow clusters when a lot of feeding buzzes are produced, there's also a lot of conspecific calls that can be heard nearby. So that's really exciting, and I really hope that we can do a bit more of that with our own bats in Panama soon. So one thing that we are doing now, and this is the last sneak peek from my group, is to look at that within a species. So instead of comparing different species with different adaptations, we have started to work, and this is a collaboration with T. Gomera and Meg Crowfoot, to look at Philostomus hastatus, which was described from Trinidad to live in very stable social female groups that are thought to be so stable, at least partly because this bat seasonally feeds on the flower and the nectar of balsa flowers. And these bats return to the colony and to their group and produce specific screech calls that are group specific to recruit their group members to this food source and then exploit it together. And that's a really interesting concept, because it would be showing active recruitment. And so Teague and I went out a couple of years ago and wanted to repeat this in Panama with GPS tags on whole groups of bats, just to see whether they really fly together and what they then do at the resource. And so when we did that, though, we didn't find what we expected at all. So we went to Panama and we tagged bats in a cave on Bocas del Toro, which is a little island off the coast of Panama, and then tracked groups of them. And even though they all went to the same patch of balsa trees, which was the only flowering balsa far and wide, so there were no flowering balsas on the island or anywhere on the mainland that we can find. But this little patch of trees in a river island surrounded by banana plantations had flowering balsa, and all of the bats we tagged flew this 25 kilometers across the ocean to feed on this patch. But they flew there individually. And what we suspect is that as this flowering patch came up, maybe one female discovered it and returned to the colony smelling of balsa flowers, and then that following night, other group members followed her. But once they had detected the patch, they didn't need to follow her anymore. And we kind of missed that moment during our tracking. And we couldn't track long enough to see them switch to the next patch. And so then they flew individually there when we were tracking them. But what it does indicate is that there's some variability in the system, also confirmed by the fact that the bat groups that we did catch here were not nearly as stable as described in the literature. So we think there might be a gradient in how dependent they are on this resource, and how ephemeral this resource is, and linked to that, the social structure of these bats. And that's something that Camila Calderon is now starting to study in Panama. And with this, I'm at the end. I just want to briefly announce Edward again, who will be joining the cluster as a cluster postdoc and studying social facilitation of migration in these African fruit bats, because seasonal variation is something that I haven't really talked about yet. But we think that these bats follow the green wave, or predict the green wave, and use their colonies as an exploded sensory network to time their migration to it. So let's keep our fingers crossed that Edward can get his visa soon and come and join us here. And then we can have him talk about this project more himself. And with this, I'm at the end and happy to take your questions.
I was wondering, with your work with Teague, so you said the first female found this, and then there's some social information, and the others are then following, and you missed that initial part. Do you have any evidence with the period of time that you're tracking of exploratory flights or something like this to, you know, find the next food source? Anything that would point to that first female?
Sorry, Lucy, you were interrupted for me. Can you say this again? I'm so sorry.
Well, you have the sort of first initial finding, and then you think social information, and then individual sort of movement out there once they've received that. I was just wondering if you had found any evidence in that work of that initial finding, or of exploratory flights or something like this.
Yeah, no, we have not. We tried to track them a lot, but I think it's the wrong study site in Gamboa, because even though resources fluctuate so much, they're still too abundant. And we would have to do this in an area where fig occurrence is natural, which is just in gaps and trefoil gaps and stuff like that, because they're a pioneer species. And we'd love to do that, but we have not. We're hoping to maybe see something like that in the Philostomus though.
Do you think that it's possible that that then is actually really very rare? Like, you know, could you speculate of how many individuals you think need to be exploratory for the whole population to exploit?
Yes, I think it might be extremely rare actually, and maybe occur in some individuals' life, even occur never. But what we find in this bat is a fairly large network, fission fusion society network. So bats have their semi-stable groups where they mostly hang out, but these groups can then split over several subgroups and exchange among each other. One really interesting thing that we found during our behavioral experiments is that they actually really like information from animals that they don't know yet. So if you offer them information from group members versus unfamiliar individuals, then they will prefer the unknown, both of them demonstrating an unknown food source. They will try out the unknown food source of the non-group member first. So maybe they're also always trying to expand their repertoire. But yeah, because we think it's so rare, it's going to be really difficult to observe it. I hope we'll hit on a study animal sometime that does it more often.
Hey Dina, thanks for the really interesting talk. I think it was really cool to see these different specializations based on the temporal and spatial predictability of the resources. Wondering, did any of these species, like the resources may come in and out in different seasons, do any of these bats go for resources that maybe have high spatial predictability or temporal predictability in one time of the year, but in another time of the year, the resource properties are changing?
Yes, absolutely. So for Noctilio, for example, we think they only use the insect swarms so strongly during pregnancy and lactation, when the females are energetically much more challenged, and they really need to make sure they don't have to invest too much time into flying and thus expend a lot of energy, but feed a lot, because in Panama during the dry season, for example, it's very windy, and over the water, they forage like a meter over the water surface. The water surface gets very choppy, and also the insect swarms get dispersed more quickly. Then they don't use this strategy, and they do solitary foraging in the forest. So absolutely. And the same with the Philostomus that I talked about last. This is an omnivore, and only seasonally, again, during pregnancy and lactation, is specialized on the balsa flowers. which strangely seems to be a very common thing in bats, that they like nectar during this time of their life cycle. And during the rest of the year, they eat insects and small mammals and so on. And nobody's tracked them during that time, but we assume that they do that alone then. So yes, very much. Or again, Lara's bats, which are temperate zone bats. And I showed the insect availability in Poland and how it's really ephemeral. These females actually migrate there in the summer, probably to exploit this resource, which is relatively more abundant. But then during the rest of the year, they migrate back and they feed on other resources here. That's an eidolon on the picture, by the way, the species that Edward is going to study in a cluster.
I have a cheeky follow-up question, if that's all right. Just on that then, you mentioned they follow the green wave and that they use social information to do this, which seems really cool. Makes me immediately think of the paper in Science from Brett Jesmer on cumulative culture in ungulates following the green wave. Do you think you might find something similar here, or is it out of the question?
No, I really don't know what we'll find, because it's quite challenging to work with individuals in this bat. But what we do see is, I mean, why we think that it might be socially facilitated is because a cumulative number of things. So one thing is that they don't really follow the green wave, but they seem to anticipate it a little bit. So they leave a certain site before it is really, the resources are exploited there. And tied into that, they don't really adjust their foraging distances depending on colony size, because these colonies can vary from like a few thousand to several million, but the foraging distance is always the same. So they really seem to pick spots that have an ideal resource availability. But at the same time, what we see in the patterns of them arriving and leaving is that they kind of trickle in a bit more slowly over maybe the course of several weeks, and then there will be a maximum colony size. And then that drops off really, really suddenly. So almost from one day to the next, the bats will leave. It's almost like there's a trigger somehow. And what we see is that few individuals of these bats will not just forage, but they will go further into the landscape, up to 150 kilometers or more in a day, sometimes even spend the night somewhere and then come back. And somehow maybe these animals are sensing the landscape to kind of look for upcoming resources elsewhere. And that then, again, this information is transmitted in the colony. And tied into that, one thing we are looking at is a behavior that we call flushing, where the bats fly up every evening and they kind of circle above the colony before they emerge in the evening to forage. And this is a main emergence colony direction for like the whole colony. They don't fly out equally all around. And this main direction changes, and maybe this also then at some point kind of transitions into migration. But we'll have to do a lot more work on that.
That sounds a bit less like culture and ungulates and more like my understanding of quorum sensing in bees. And maybe it's some immediate.
Exactly, yes, something like that.
Cool.
Yeah, thank you. I like it too.